Alright, this video is your cell transport Prelab. There will be a link on the website that has this Google Doc on it. I'll probably share it so you guys can access it. It's pretty much just kind of an overview of the same stuff that we talked about in class with passive transport. Okay, I would like you and active transport according to the video, because I did put a video for active transport in case you guys want to take a look at that. And then, so you're going to go ahead and read through this. Okay, it's pretty straightforward. Like I said, it's kind of a refresher. And then on the second page, it talks about what you're actually going to be doing in the lab. Okay, so in this lab, you're going to be exposed to 3 different experiments. Okay, experiment 1 will be done to determine how cell size affects the diffusion of materials. Okay, you're going to collect quantitative data. All right, now how you're going to be doing this is you're going to have, cut up 2 different sizes of potato. Okay, and then make sure you get the measurements of it so you can calculate the surface area and the volume, and then you're going to submerge it in a dye, crystal violet, for 24 hours in the incubator, and then tomorrow or the next day, you will look and cut the potato in half, cut that cube in half, and measure how far the dye has diffused in. Things you should keep in mind is what is that dye representing in this model, and how does surface area and volume ratio affect how the cells get nutrients and stuff like that. So important, you will submit 1 piece of paper from your lab book, okay, because you want a record of it, with a data table and calculations on the top half of the page and the graph on the bottom half, and it should take up the whole bottom half of the page. So please make sure that you have a whole half of a page of graph. Okay. Experiment 2 will be done to determine how membranes are selective in the passage of materials. Okay, this is a qualitative lab where you're going to use biochemical tests. You're going to use a Benedict's test, and you're going to use a Biret test for proteins. You know what the Benedict's test is for determining reducing sugars, but you're going to use and you also use a starch test to determine or iodine test to determine starch. Okay, I'll have some positive controls set up for you so you know what to look for. You'll just set the labs up tomorrow, and then the next day you will see your results and come to conclusions. Okay, experiment 3 that's not what I wanted. Okay, experiment 3. There we go, looks a little better. Experiment 3 will be done to determine the water potential and solute concentration of different types of potatoes. You're going to have sweet potato and you're going to have a russet potato. Okay, you will be designing an experiment to determine these characteristics. Now, if you're wanting to know what water potential is, I am also going to put on a video for water potential from Bozeman Science, so please make sure you take a look at that. But the materials that you'll have available to you, and you will need just these materials: sweet potato, a russet potato, a French fry cutter, which I'll probably have the potatoes already cut up for you. All you'll have to do is just do the secondary cuts, if that. I may have those done for you as well. A metric ruler, the triple beam balance, some different sugar solutions, and plastic cups. I really forgot to put cups. So plastic cups, and then you're going to put the potatoes in the cups. The concentrations of sugar solution you'll have is a 1.0 molar sucrose solution, a 0.75 molar sucrose solution, a 0.50 molar sucrose solution, and a 0.25 molar sucrose solution. And there will be 1 more, because you're going to have 5 cups, or actually you're going to have 10 cups, because you're going to have 1 cup per potato. I also want you to have some distilled water, which hopefully you'd realize is 0.0 molar sucrose. All right? So, like I said, watch the video on water potential, and you guys will, if you need some help, in your designing, you're more than welcome to come in at lunch, and I can point you in the right direction. But hopefully, this one's straightforward enough that we can determine that. Okay. If you have any questions, you're welcome to email me as well.